Hello, my topic is enzymology of DNA replication. We will be studying about both prokaryotic and eukaryotic in this slide. Enzymology of prokaryotic DNA replication. This happens in the prokaryotic cells, that is the unicellular organisms. We will be seeing about many enzymes in these coming slides which are required for proper DNA replication. The first one is the DNA polymerase. There are 5 types involved in the DNA replication. The first one is the DNA polymerase alpha. This is the largest type in the DNA polymerase family, and it has 4 subunits and is thus called as a tetramer. These 4 subunits are Pol A 1, 2, 3 and 4. The main function is polymerization activity. The second one is the DNA polymerase beta, which is the smallest of all the DNA polymerase types. It is only found in the vertebrates but not in invertebrates. It is also found in the nucleus of the cell but nowhere else. DNA polymerase gamma is the third type, and it is involved in the DNA replication in mitochondria. This has 2 subunits and is thus called dimer, encoded in the nucleus of the cell, but it is not present in the cytoplasm or anywhere else. The fourth type is the DNA polymerase delta, which is the main replicating enzyme which is involved in the DNA replication. It has 2 subunits and thus called the dimer. It is encoded in the nucleus. DNA polymerase epsilon is the 5th type, which is also encoded in the nucleus, and it has 4 subunits and is called a tetramer. This is involved in the synthesis of the leading strand. A leading strand is a continuous strand. The second type of enzyme is the pCNA. This is also called as the proliferating cells nuclease antigen. It is involved in this DNA synthesis and repair mechanism as it acts as a clamp. The third type is the helicase enzyme. Helicase enzyme helps in the unwinding of the double-stranded DNA into the single-stranded DNA at origin. This process is energy requiring, that is, it will take ATP. It will use ATP in this process, and it is also a high temperature consuming process because the DNA has to denature. SSB proteins. This, after helicase unwinds the DNA, SSB proteins bind so that the DNA should not wind back. There's a small mistake. This is wind back. Okay. So SSB protein is a single-stranded binding proteins. These will bind to the single-stranded DNA, and it will prevent the DNA to not wind back, so that the replication can go smoothly, and this prevents renaturation, so that the process is not stopped. It is also a stabilizing protein which stabilizes the ssDNA. The next one is the topoisomerase. There are 2 types, type 1 and 2. It helps in the removal of the topological stress due to unwinding of the DNA, and also it takes precautions that the ssDNA, which is the single-stranded DNA, will not supercoil. Then is the RNA primase. It synthesizes the RNA primer for the replication process. It works with DNA polymerase alpha, which is the main replicating enzyme during the replication. DNA ligase. At the end of the replication, there will be 4 locations which will not be filled with the nucleotides. So this will become a lagging strand, and so these gaps will be filled with the help of the DNA ligase enzyme. This DNA ligase enzyme will join the different leftover parts of the DNA chain. Thus, it helps in the DNA repair mechanism. And the last one is the flap endonuclease. The nucleus. This helps in the removal of primer on both leading and the lagging strand. Once the primer's work is done, it has to be removed. So the flap endonuclease enzyme does its job. Then we'll move on to the eukaryotic enzymology DNA replication. Only the names differ. The names of the enzymes differ. All the other part, the process and the functions remain the same. The first is the tag 1 and tag 2, which help in the unwinding of the double-stranded DNA into the single-stranded DNA, this process is energy consuming. It consumes ATP. These two enzymes break the hydrogen bond and helps in the separation of the strands. Then is the DNA polymerase. Unlike the prokaryotic DNA polymerase, this has 3 types. The DNA polymerase 1, it is the first type of DNA polymerase which is discovered by Comberg. It is encoded by gene polymerase A and consists of 928 amino acids. Then is this DNA polymerase 2. The main function of the DNA polymerase 2 is the polymerization of the DNA. It is a proofreading. It does its proofreading activity, which means whether all the nucleotides are encoded properly in the chain, or if there are any mistakes in the DNA replication. Then we will move on to the DNA polymerase 3. Its main function is the polymerization of the DNA in the eukaryotes. It is a haloenzyme that is composed of 10 distinct proteins, and it has 3 functional molecules and are separately responsible for their actions on DNA replication. Okay, next is the RPA or the RFA enzyme. This is also known as the replication factor A. It has the same functions as the SSB proteins, which is the single-stranded binding proteins, and it also plays an important role in stabilizing single-stranded DNA. The fourth one is the RFC, which is the replication factor C. It acts as a clamp reader. It also plays an important role in linking between the DNA polymerase alpha and the DNA. MF1 enzyme plays an important role in the DNA replication, as after the replication the RNA primer has to be removed. So the main function of this enzyme is the removal of the RNA primer from the leading strand and the lagging strand. These are my references. Thank you.